Hello and welcome all of you in my YouTube channel, and I'm back with my new video. And today we are going to study about a very important topic, or I would say a very specific topic for clinical laboratory, which is lab diagnosis of tuberculosis. So how we detect the diagnosis of tuberculosis and how this mycobacterium tuberculi, which causes tuberculosis, is very effective in all the ways that we are going to study today. So now, without wasting any further time, let's get into the video. So laboratory diagnosis of tuberculosis involves demonstration of the important bacteria, that is bacillus, in the lesson of microscopy, that is a technique isolating in the culture by transmitting the infection to the experimental animals and demonstrating hypersensitivity to tuberculoprotein using molecular diagnostic methods. So a lot of methods are used here, like with the help of microscopes it is identified, and using my molecular diagnostic methods. And some of the methods that can help in identification are really involved in these diagnoses. Laboratory diagnosis of tuberculosis also includes different type of steps that help in the identification of the process. First is collection of specimen. So in the first is pulmonary tuberculosis, in which sputum collection is done in a clean wide mounted container, and then 2 sputum specimens are spotted in the same visit. And in the same way we found laryngeal swab, bronchial washing may also be collected in sputum. In the case of children's, we collect sputum gastric lavage, that is examined. If I talk about in extra, in the part B, pulmonary tuberculosis, EPTB, vary depending on the size, and lymph nodes, aspirates, pleural fluid, urine, sinus fluid, cerebrospinal fluid, CSF, pus from cold abscess, and tissue biopsies are some of the collection methods. Now come to the digestion, decontamination, concentration of specimen. Now specimens are non-sterile sites as they need prior treatment for digestion to liquefy thick pus cells and homogenization. Decontamination is also to inhibit normal flora. Concentration to increase the yield, and processing to be done in second class biosafety cabinet, because as I should tell you, tuberculosis is a very effective disease which can lead to death. So it's very drastic and severe. So you have to keep in mind that you have to take a lot of precautions while dealing with this. Petrikov method, sputum 4% NaOH, equal volume of incubation 37 degrees Celsius at the temperature, and shaking till become dear. And these centrifuged at 3000 rpm for 20 minutes and then neutralized and indicated in phenol red, used for smear culture inoculation. Now if I talk about the absorption, and means the digestion part, decontamination, concentration part of the specimen, NaLC, N-acetyl and L-cysteine plus 2% NaOH, better the Petrikov NaLC, liquefies the sputum, and NaOH kills normal flora. The sample is also neutralized in this case. And also the most important part is the culture is automated system, and a smear microscopy is used in this, but not useful for the culture point of view. Next come the direct microscopy by acid fast staining. So with the help of Ziehl-Neelsen staining, that is Zn staining, direct concentration of smear of sputum is examined, and sputum is reliable single method for diagnosis. Smear are prepared for thick purulent part of sputum, dried, heat fixed and stained at Zn technique. Zn AFP report says as INTCP guidelines that this, what you see on your screen right now, is shown as the mycobacterium tuberculosis bacteria, which causes positive results, and it shows a lot of in acid fasting. Now comes the laboratory diagnosis of tuberculosis. When growth is done on LJ medium, okay, LJ medium is Lowenstein medium, and in this medium rapid growth is seen in 7 days. Also in McConkey also the same, but somehow the Mycobacterium fortuitum complex and telluride reduction is seen. Mycobacterium phyllae and smugognostis, sometimes in large, light, dark, and no pigment, as the two groups you see on your screen: photochromas, secrochromas, and chromogens. And when it's a slow growth, so in the case of slow growth, you see niacin and Mycobacterium tuberculosis pigments and scanty smooth flat colonies, while the identification and there is no pigment. And when it is negative, the BCG, and when it is positive, Mycobacterium bovis. So this is the diagnosis of tuberculosis. Very easy, very precise. The slides I have used are very brief, so that you are able to identify and able to understand it, and it is very clear to you that how we diagnose, how we collect, how we isolate, and how we are going to diagnose, because that is really important in tuberculosis. Handling procedure, working with that is really a very difficult task to perform, because tuberculosis, as I said too many times, is a very difficult bacteria and a very communicable bacteria, which can cause tuberculosis and lead to severe disease. So yes, thank you so much all of you for watching, and like, share, and subscribe to my channel. I'm helping you each and every day with the new videos related to microbiology and biotechnology. I hope you like this one also. This helps you in understanding the disease and the bacteria itself. And thank you so much for watching. Share with your friends so that they can be able to understand it easily. Thank you so much.